pleasure for me to introduce John Lowengrub as the second speaker this morning. John comes from the University of Irvine in California, and he's an expert in modeling, numerical analysis, mathematical analysis of models, also the applications, and I'm sure we are going to get a glimpse of every of his beautiful skills, and look forward to his talk.
Thanks, Alf, and thanks very much to the organizers for inviting me. And so I'm going to talk about how one models, well, normal, but I'm going to focus on tumor growth. And in particular, I'm going to talk about interaction of the tumor with the host and focusing on the vascular system. And then what we want to use is the mathematical models to predict outcomes of therapy to help direct experimental and clinical investigations. And so this is joint work with a number of different people. So two of my students, Huaming, who just graduated from math, and Monica Romero, who's in biomedical engineering, is going to graduate next year. And this is actually her model system. So she's had a math background, but is now doing experiments. And so she's jointly supervised by Chris Hughes, who is an expert in angiogenesis. And what she's doing is she's growing tumors in a microfluidic device with blood vessels. And so ultimately, we are planning to use this model system to test some of the predictions that I'm going to give you today in the modeling. And so this is a particular example of a colon cancer with a blood vessel systems that you see in red. And so they're grown, the tumor and the vessels are grown together in a microfluidic device. So basically just a little bit to start about cancer. And so cancer is really the loss of control. And so the cells and tissues in the body are tightly regulated through feedback, both chemical and mechanical. And so cancer is when this feedback system gets out of control. And so here's an example of just a cartoon of how cancer could develop from epithelial tissue. So these are basically tissues that line the organs and separate, let's say, organ from stroma. And so simple epithelium basically says you have one layer of cells. You could have stratified epithelium where you have multiple layers of cells. This would be, for example, like part of the colon. And then there's a basement membrane and surrounded by stroma where you have blood vessels, extracellular matrix, and so on. And then as a series of mutations can occur, or that could change essentially sensitivity to feedback, or basically losing growth inhibition feedback, then one can, if you have a series of them, then one can, the cells begin to grow abnormally. And so then the simple epithelium can have a pileup of cells, and these cells then can degrade the basement membrane, separating this epithelium from the stroma, and then they can invade the stroma where they can get access to blood vessels, penetrate the blood vessels, be transported to other organs, usually the next one down the vascular system, or they could get into the lymph system, and then metastasize. And so at this point, this is when cancers are usually observed because you have symptoms, but at this point, it's the most dangerous. And so what's really regulated, of course, is the cell number. And so, which is basically the number of birth events minus the number of death events. And so for tissues, this change should be, at least in adults, should be very minimal. The traditional view is that cancer is really a condition of the cell cycle. So cells could essentially not be stopping when they should be, and so then they could dysregulate their basically the birth and death cycle. And so that's an example of direct control. So the cells are going through the cycle and not stopping when they should. But there's also, so how can that manifest itself? Well, that could manifest itself because what really is happening in tissues is that there is feedback. And so feedback is how the systems are tightly regulated. And so you could imagine that instead of just controlling how cells go through the cell cycle, you could control on you could allow them still to go through the cycle, but you could change, for example, the way that their fate after they go through the cycle. So, for example, here's an example of a lineage of cells where basically we start from a stem cell. The stem cell will divide. It has the chance to produce another stem cell or produce a more differentiated cell. And so we can identify the different fates with a couple of parameters. So we can say P is the probability of remaining a stem cell after division. And V, we can talk about division rate. So 1 minus P is the probability that they differentiate. And so the stem cells can self-renew and they can differentiate into more differentiated cells. And then these more differentiated cells, typically called committed progenitors, they then have some degree of self-renewal as well, and then they can also differentiate into, there could be any number of these, and they can differentiate into terminally differentiated cells. And one can look at this as a system, as a dynamical system, and ask under what conditions can you have the nice, what one would expect to be robustness in terms of biological functionality. So, for example, under what conditions do you have nice homeostatic equilibrium, conditions that are nice and stable? Can you have, if you were to have a wound, so you get rid of some of these cells, how quickly can the system regenerate, and will it always grow to the same size? And so this work was led by Arthur Lander, who's a developmental biologist at UC Irvine. And what they discovered is that what's critical to homeostasis is control not necessarily on division rates, like what I was describing at the beginning with a direct control, but rather indirect control where the feedback is actually on the fate. And so if one introduces negative feedback on the fates, in other words, have the differentiated cells produce factors that negatively feedback on these self-renewal probabilities. So it's kind of counterintuitive. The more differentiated cells you have, the lower, this is because this is negative feedback, the lower the self-renewal. And it turns out that that is the feedback that's critical in this type of system to having a very robust dynamical system. And then there's evidence in the literature, and also that these guys found for specific tissues, that these type of negative feedback on cell fate is an action of the so-called transforming growth factor beta superfamily. So there's a superfamily of proteins that do exactly this. But in addition, there can be positive factors. So these are negative factors typically, but there can be positive factors that increase these the self-renewal probabilities. And so examples of this are just listed a few here: Wnt, fibroblast growth factors, endothelial cell growth factors, Notch, sonic hedgehog, so on. And so this work gives insight because it basically says, well, it gives insight into what's stability for this dynamical system if one writes down a system of ordinary differential equations for the populations. But it also gives a new interpretation for behavior. And so what may distinguish a cell, one cell type from another cell type? So we usually think of the stem cells as always self-renewing. But in fact, the behavior may not be intrinsic, but may be controlled by the environment. So that the difference between this cell type and this cell type may actually just be the signals it's feeling. So this could be perfectly capable of self-renewing, as the stem cell, but if it's under the right conditions, that increases this value of p, let's say above 1/2, so that more than half the time you would stay at the same cell type. Yes?
What's the CP and PD?
So committed progenitor and terminally differentiated. So now this is normal. And so one can ask, well, is this relevant for cancer? Because in cancer, one thinks generically that all the cells are capable of dividing, all the cells are capable of recapitulating a tumor. And the question is that really true? And in cancer, we have a loss of control. Is there any remaining control in the system? Should one even think about this type of model in cancer? And so there's experiments.
Is it really right always to say loss of control? Wouldn't it be more parsimonious to say change of control?
Yes, and so that's what I'm going to motivate here. Right, so the convention would be thinking that there was loss of control, but actually I'm going to show you evidence that control is not fully lost. It may just be different. So, so there's now been a variety of experiments that show that this progression and control actually exists in cancer as well, just different from normal. So here's an example of breast tumors, so the BRCA1 breast tumors. This is in a mouse model, as you can see. And so basically what these experimentalists did is they take these tumors, these tumor cells, implant them in a mouse, and they'll generate a tumor. Then what they do is they remove the tumor from the mouse, and they identify the putative cancer stem cells, so by their surface proteins. And so for these types of cells, they identify the cells with the cancer stem cells, and I'll say more about what those are in a second, with this value being high and this value being medium, so they call them medium high. And so what a cancer stem cell is really a functional definition. I'll give a bit more insight into that as we go along. But essentially, these are the functional definition of a cancer stem cell is basically a cell that, when you put it into an animal, will create a new cancer. And so it's somewhat like a stem cell, but not necessarily the same. May not have the same degree of pluripotency. Whether they came from normal stem cells or not, still open questions. But in any case, for this particular tumor, about 5% of the cells in the tumor have these markers and are the putative cancer stem cells. So then what they do is they purify basically they sort the cells. They take a 99.99% collection of cancer, what would be called cancer stem cells. They implant that 99.9% number of cells back into the mouse. They wait a while, creates a new tumor. They remove the tumor, sort the cells, and find that the number of cells with those markers went back down to 5% again. So this percentage of cells with the cancer stem cell phenotype, at least as defined here, is fairly robust. And so that we take that as an indication that there still is some type of feedback remaining in the system. Now, of course, you can adjust that, and that feedback may change over time, right, as cancers progress and may become less and less differentiated, so less and less responsive to negative feedback. And so here's an example of how you can manipulate the stem cell fractions just by treatment. And so, in fact, traditional treatments actually, in a sense, do the wrong thing. They select for the cancer stem cells. And so here's an example of treating these mouse with cisplatin, a typical chemotherapy agent. And so what happens is that, again, the original tumor is a roughly 5% cancer stem cells, and after a few cycles of treatment and reimplanting the cells, you can crank that up to 20%. And now why is that bad? Well, of course it's bad because these guys can generate new tumors, but not only that, these cells, these so-called cancer stem cells, have better, are much better at resisting therapy. They have better pumps to get out toxins. They have better DNA repair mechanisms. And so these dramatic increases in population of these cells can lead to enhanced resistance. And this is one mechanism for the increased resistance that one observes as you treat a tumor. So basically that leads one to think about what's the so-called cancer stem cell hypothesis. And so this hypothesis basically says that, well, now there's been these so-called cancer stem cells have been now found in a number of different tumors. There's a few listed here. It was first discovered in leukemias, then breast, and so on. And so the idea is that the tumor is filled with many cells, but not all of them have the capability of sustaining the tumor. And so the tumor progression and response to the treatment is really controlled by this small population of cells with these phenotype. Yeah.
Is there something which I would have to understand as non-differentiated and well-differentiated cells, or?
So the idea, the picture is that one doesn't necessarily know the degree of differentiation in the tumor, but that they don't have this so-called markers that indicate this population that's capable of reproducing the tumor. So there's only a few cells with those markers that seem to be capable of reproducing the tumor. So I didn't mention here before, but now if one goes back, oops, I'm going the wrong way. If one were to go back and say, now let's take some of these cells and implant, they won't form a tumor. At least not with the same degree of probability. Much lower probability.
What's different between these two types?
These cancer stem cells are the ones that seem to be capable of self-renewal. So they're more like the early stage part of the lineage. They may be the stem cell-like or committed progenitor-like, but they're capable of self-renewal, whereas these cells are not.
And generally they don't senesce, although it's not 100%.
Right, right. So they may not, they may always have that proliferative capability. But those are all good questions. It's not completely understood. Okay, and so that's roughly the cancer stem cell hypothesis. So now, the cancer stem cells also can enhance invasiveness and aggressiveness. So one example that I'm going to keep coming back to is glioblastoma, which is a very aggressive brain tumor. So it's highly heterogeneous, very highly vascularized, and very poor prognosis. Usually, when these are diagnosed, the survivability is on the order of a year. There have been some newer treatments that may be extending it further, but it's a very devastating disease. And so, one can, if you look inside the tumor, then one can find, well, there's the cancer stem cells, there's a mix of endothelium, parasites, all different types of cells in the tumor. So it's highly heterogeneous. And so to give an example of how the stem cells can play a role in the progression of this disease, one can do a mouse model, so that was done by these guys, where they implant glioblastoma cells into the brain of a mouse. And so what they did is they varied the percent, so the CD133 is a putative stem cell marker for glioblastoma stem cells. And so they take their initial collection of cells that they implant into the brain, has varying degree, essentially percentages of those cancer stem cells. So if they don't implant any cancer stem cells, tumors don't form. So they're implanting roughly the same number of cells in each type. They're just varying the percentage that are cancer stem cells, at least with these markers. So as they start to increase the percentage, you can see that the brain actually turns redder and redder. And that's because of the interaction between the stem cells and the vascular system. So the redness is from the blood vessels that have formed, and so the brains are all roughly the same size. They have to essentially kill the mouse when the tumor gets to be big enough size. But you can see with this 8%, now the brain is all red, so there's a very strong angiogenic response. And actually, if we look at the days till development of these tumors as a function of the initial stem cell percentage, basically it takes about 50 days at 2% for the tumor to be observable, at least neurologically. If you increase that to 4%, it's down to 30, and 8% is down to less than 20. So it's a dramatically increased proliferation and angiogenic response. And so one can look, at least for glioblastoma and many other types of tumors, one can look at the signals that are playing a role. And so they're roughly divided into signals that are enhancing self-renewal and signals that are in upregulating differentiation. And so, there can be overlap. For example, TGF-beta can be, depending on which superfamily members you're talking about, can be promoting and also inhibiting. And likewise, Notch can do that as well. And so, so I'll talk about, at least I'm going to focus mainly on one of them, but these types of signals generically are conserved in other types of cancers, maybe some playing a more of a role than others, but generally you have the same type of players, pro-self-renewal and anti-self-renewal. So just to talk about one, to give you an example, could talk about Wnt signaling. And so Wnt is a pathway, basically. So there's a protein essentially that will bind to a receptor. It has inhibitors that can prevent it from binding, but once it binds, then it can trigger a cascade, leading to essentially translocation of beta-catenin in the cell to the nucleus, and that can produce a whole long cascade of events. In particular, it's found that Wnts are produced by cancer stem cells. So in tumors, Wnts are produced by cancer stem cells. In fact, in a variety of tumors, the cancer stem cells can be targeted, or at least identified by their level of Wnt activity. That's particularly true in colon. And that, as I mentioned, there's these inhibitors, and there's a few examples, so this is one that I'm aware of, where it can be shown that the stem cells can produce both the activator, the Wnt, and its inhibitor. So the same cells can produce both. And so if you have the activator inhibitor, and what's known is that Wnts are very sticky, so they don't diffuse very far. The inhibitors are much less sticky. They can diffuse farther, and so that can suggest a reaction diffusion type model or a Turing type model, which then, as we know, Turing models have the potential to form patterns that could then potentially create a stem cell niche. So we'll see how that plays out. And what's also observed is that Wnt signaling is very heterogeneous spatially. So, for example, in colon cancer, where I have at least this figure, all the cells are constitutively upregulating Wnt signaling, but if one searches for the actual levels of Wnt signaling, so genetically they should be upregulating Wnt signaling, but the actual levels of Wnt signaling, as indicated here by this green fluorescent protein, are heterogeneous. And there's also examples of the heterogeneity of its Wnt inhibitors, and so in a variety of different tumors, one can find, for example, here this inhibitor is in green, so it's non-uniform, and it's actually at the boundary. And one can find it also in the epidermis. So given this background that there's heterogeneity, spatial, there's lineages, now how can we develop a mathematical model to try to integrate and also together with the vasculature, because I showed you that the stem cells can be affecting the vascular response. So how can we put things together and try to model this mathematically and then try to gain some insight from the model? So there's a whole bunch of ways one can do it, and I'm going to talk about one particular way, which is using continuum models. One could use discrete models as well. And the continuum has the advantage that we have pretty fast solvers for it, and that we can deal with larger systems. So I'm going to give a particular framework for doing this, and so we've used this framework in a number of different circumstances. So we've also used this framework for looking at developmental systems and morphogenesis, but I'm going to talk primarily in terms of the tumor context today. And so the idea is that, well, if we have a bunch of cell types, then let's introduce volume fractions for each of the cell types. So that means at a point in space, there's some fraction of the volume that's one cell type or another cell type. And so we can introduce these fields that are volume fractions. So for the viable tumor cells, we can track the dead cells, and so the total tumor is the sum of the viable plus dead. One can track any number of host cells, and one can track the liquid, the water that's needed, that the cell should be uptaking. And so then one just writes down the simplest idea, which basically is a conservation equation for the volume fractions. So there's a density associated with the cells. There's the cells can be transported by a velocity, and there's a flux, and then there's source terms. And so for the model I'll show, we'll make a simplification where we can just assume the cell, so that the water kind of moves around as it has to for balance. So we'll assume the water fraction is constant and the solid fraction is constant. So that's pretty good approximations away from highly necrotic regions. And essentially this allows you to decouple the solid and liquid equations, so the solid then will provide a source to the liquid. So you remove the direct coupling. It's not essential, it just makes the model easier. And so now what we'll do is we still have to, we can make those assumptions, but we have to then, I have to tell you then how to fill in these terms. So basically how to determine the velocity, how to determine the flux, and how to determine the source terms. And so what we'll do is we'll use an energy variation argument to try to get velocity and flux. And so the idea is that if we have cell type I and cell type J, they can interact. And so this Jij is the interaction potential between this type and that type. And so that could describe, for example, repulsive and attractive interactions. And then if one assumes that these are sufficiently localized, so in other words, the scales are small, one can derive a gradient expansion of this. So then you have a leading term with where the non-locality here, so this is an integral, this is a double integral over x and y, but then one can reduce it basically because things are non-local, it becomes weakly non-local and depending only on gradients. So that has an advantage in terms of the cost of the computational algorithm. And so then what you can do is follow statistical physics and basically calculate the time derivative of the energy, and then we can simply choose fluxes that are consistent with the energy being non-increasing. And so that would be true if we're not allowing proliferation events, because then that's outside the system. And so if you follow this formalism, then one can identify fluxes with variation of the energy with respect to the volume fractions. And so this is the variational derivative. You use variational derivative because your energy depends on gradients. So you just kind of follow your nose on how to do that. But then the overall system is conservative, so the flux all have to balance. So you get to choose, if you have n components, you get to choose n minus 1 of these, and then the last one is determined. Yes?
I mean, that would be, I would be perfectly fine with that if that would be not an active system. I mean, it's energy consuming system, so you don't know whether you're following the free energy gradient.
That's true. And so what we're not, what we're assuming here is that suppose that the proliferation were very low, and suppose death were very low, then what would your fluxes be in such a system? And then we're going to add that, so then we're going to use that as a basis and then add back in the proliferation and death. And so overall, our system won't satisfy this once we allow proliferation and death. But you're correct that if we had proliferation and death, maybe those are going to be indirectly affecting these fluxes, and that's possible.
But even the fluxes can be something with
Correct.
active
So, and that's possible. Yep.
It is not an in, right?
What's that?
That is not in.
That's correct. That's not here. And so one can then analogously get the velocities. And so here we can take a very simple law, which is Darcy's law. So we're essentially treating the tumor as a fluid. And but one can, again, make more complicated rules. So, for example, one can use elastic materials and generate velocities using an elastic energy. So that's kind of complicated. And the reason we do this is because it's fairly simple. And so we can write down a fairly simple model that has the right, what we believe are the right features. And so to make the model more definite and to get something that's a bit more tractable, then we can make the following assumption that these tumor cells prefer to be together. The different types of tumor cells might mix, but they want to be separated from the host, at least with some degree. And so a simple model that describes this would be to say now our energy just depends on the total number of tumor cells, so phi t, which is the sum of the viable plus the dead. And as our kind of motivating example, this work on essentially separation of neural and epithelial cells, so these fundamental experiments done years ago. And then what we can do, if we imagine that they want to be totally separate like these cells, we can have an energy that, so this F is, we can use as a double well energy. So this energy would say that you want, you're going to have minimum energy where phi t is 0, so where you have host, or where phi t is equal to the solid fraction, so you have only tumor cells. One can describe the mixing between these two by relaxing that, by changing this function. So if you had a single well, then the two would want to fully mix. And the methodology would be similar. And so we can also assume that the cells move together. So that's consistent with our assumption that let's say the total solid fraction is constant. And so then if they're going to move together, we can describe their movement with a mass average velocity. So all the cell types, the solid cell types would move with the same velocity. And then that velocity, we then can follow our simple formalism to relate that to the gradient in pressure. And so basically the idea is that as cells are dividing, they're creating pressure. Pressure changes are creating motion. And that's a passive motion. One can also add active contributions from, for example, haptotaxis that Alf talked about earlier, but also with chemotaxis and so on, additional forcing. And then one can get and then using this as the energy, one can get the simple Fick's law for the fluxes, where the flux is proportional to the gradient of the chemical potential, where the chemical potential is the variational derivative of the energy, which for this system takes this form. And so ultimately, then putting that all together, one has a conservation equation. If we assume also the densities of the different cell types are the same, again, all just simplifying assumptions to get something that's tractable, then this is what the system looks like for the different cell types. So there's the velocity, which is related to the gradients of pressure, and then here's our fluxes, and then the source terms, which I have to tell you about. And so what this ends up being is at least for one of the cell types, so for example, if we add all these up, we get an equation for the solid component phi t. And then that equation for phi t looks like a Cahn-Hilliard type system, more generalized Cahn-Hilliard system. So it's fourth order nonlinear. And so now the next piece is to talk about the source terms. And so for the source terms, now we go back to the biology, and then we go to this, our lineage picture again, and then we want to relate the source terms to our lineage. So again, we have the stem cells, the committed progenitor cells, and the terminally differentiated cells, and various feedbacks, which I'll talk about as we go on. And so for the source terms, basically now we have to describe how the system is going to evolve and change the balance of terms in the various balance of mass, essentially, in the various places. So for the stem cells, well, what can they do? They can renew or they can differentiate. And so we're going to assume that will occur upon division. So there's the division rate, now I call it lambda. And the division rate, will, as a very simple assumption, we'll just assume that is dependent on the levels of nutrients. So we'll just have a characteristic nutrient field, which is some combination of oxygen, glucose, growth factors, and so on. And then P0 fraction of them will stay at stem cells, and 1 minus P0 will differentiate. And so the P0 minus 1 minus P0 is 2P0 minus 1. And so P0 is this essentially probability or fraction of cells that are staying stem cells. Then for the committed progenitors, well, the differentiated ones produce 2, so you get a factor of 2. So this comes from the differentiating stem cells. And then this is the self-renewal of the committed progenitors. And then the differentiating committed progenitors produce 2 terminally differentiated cells, and we'll assume that the only thing those terminally differentiated cells can do is die. And so now, how does feedback come in? Well, feedback comes in because that's the P's, okay? And so I've written it here for P0, but the P can have feedback from a negative factor, and that's what I talked about for robustness for the dynamical system, and a positive factor, which can be upregulating self-renewal. And so here, this negative factor, so this is a concentration, and then this guy of this negative factor, and that guy is being produced by the terminally differentiated cells. So biologically, it's known that these tend to be the sources of the negative factors. The positive factor, that guy is we're going to assume is produced by the stem cells. Could be produced by a combination of these guys, but for simplicity, we'll just assume it's the stem cells. And so these guys are producing their own positive factor. Then, of course, to complete the picture, we need to know how the nutrient will evolve. And so the nutrient, we're just going to take a very simple equation. We'll take a steady state equation. So it's really quasi-steady because as the tumor changes, as time, because the tumor cells are time dependent, then the source terms change, so that changes the nutrient. But at each step, the nutrient equilibrates, and that's because the diffusion is occurring much faster than proliferation. And so nutrient will diffuse, it will be uptaken, and can be provided through the vasculature. Now, to complete this, I have to tell you about the signaling, right? So these factors. And so for these factors, then we're going to pose essentially reaction diffusion equation. So for the positive factors, for example, for Wnt, if we keep that in the back of our minds, then what's known is that Wnt signaling kicks off a cascade of other signals that have in part an effect of increasing the signal, increasing Wnt signaling. So it can nonlinearly upregulate itself. So that's F. So this F has a nonlinear production term from the Wnt itself. And then it has inhibitors. And so a simple way of modeling the inhibitor is using the Gera-Meinhardt model. And so an implicit assumption here is that the Wnt is upregulating its own inhibitor, and that's doing it through the stem cells. And also, one needs certain levels of nutrient in order for that to occur. The cells have to have enough food. Then, of course, decay, and there can be a source. This is not strictly necessary. And then the concentrations are transported by the water. And then the diffusion coefficient for the this activator, the positive factor, is small, and this is larger. And so that's this is the positive factor. This guy is just affecting the positive factor, the positive feedback factor. And then we have to talk about the negative factor. And so then the idea of the negative factor, again, we'll take a quasi-steady equation. One could also make it time-dependent, but these tend to be rapidly diffusing. And so again, it's an equation very similar to the nutrient. It's just a uptake, diffusion, and then the source of this negative factor is from the terminally differentiated cells. So the sources will be spatially, temporally varying because the distributions of cells vary. So now if one puts this all together, then you have a big system you have to solve computationally. And so part of the reason for our simplifying assumptions was to get to a problem that we can tractably solve. Yeah?
How do you couple this? Do you couple this velocity to the velocity for the cells?
Yes.
Exactly
So yes. So so basically, I didn't show you the water equation. So, but we can take a Darcy law for the water equation, and so water is being uptaken by cells. And so we know the source term, and that's related to what the cells are doing. And so basically the cells, as they're proliferating, they're uptaking water. As they're dying, they're releasing water. So you know the source term in the water equation, and that allows you to determine the water pressure. And then once from the a simple Darcy's law for the water pressure, so the velocity is proportional to the gradient of the water pressure, the water velocity, then you know these terms. And so they're coupled.
And also, is it possible, do you have a picture of where these models are posed, how they are posed, in which domains they are posed?
You know, in terms of under what conditions do we have well-posed problems? I don't have theory to show that it's a well-posed equation, but I'm very confident it is a well-posed system. But it's not so easy to analyze. One can analyze simpler versions of it. For example, you could look at a Cahn-Hilliard Healy-Shaw model or Cahn-Hilliard-Stokes type model where you couple the Cahn-Hilliard with a fluid, and those are well-posed. And so now this is additional on top of that. But the basic part is well-posed. So now so we've constructed a system that we actually can solve. And so we're going to solve it in 3D. We'll use adaptive mesh refinement to get efficient solutions. And then we use, because at least one of the equations is fourth order, we have to use implicit methods. And so we use a multigrid method to solve those implicit systems efficiently. So there's a lot of technology that goes into solving the equation. But that's kind of a different story. I want to focus more on the biology and the results. So the first thing we can do is say now, suppose that we have an avascular tumor, so there's no blood vessel system, what would this do? And so this, we're taking as initial condition this kind of perturbed sphere, and we're taking a small number of cancer stem cells that will be in red. You don't see them because their threshold at which we're plotting it is higher than this. And then the green, which you're not seeing, is, again, for the same reason, will be the committed progenitor cells. And so we start with something that's uniformly distributed throughout, and then we let it go. And so you can see the green it forms in the center. The red are these regions of cancer stem cells. They form at the boundary, and they actually will drive the invasive fingering. And in this case, the tumor will actually break up and become multifocal with little regions around these cancer stem cells. And so what happened is that the system self-organizes. And it self-organizes because the stem cells are producing the positive factor as well as its inhibitor. And so that's a Gere-Meinhardt system. And so that has the potential to form patterns. And indeed, it forms the pattern. So where you have this high positive factor, you have high self-renewal, and that's what drives these clusters of stem cells. They're forming at the boundary because the production of those factors depends on the nutrient levels, and so if you tend not to form them, at least for this system, as I'll show, I'll talk about how that changes later. You tend not to form them in the interior because there's lower nutrients, so there's also less proliferation. So it's harder to generate these guys
What
inside.
makes them break up?
So what's happening is that the cells behind, so the cells back here, they're the terminally differentiated cells, they're dying. And they're only being replenished by the stem cells here. So the stem cells are dividing, and they're leaving behind the more differentiated cells, the committed progenitors in their wake. And they tend to divide more here than here because the nutrient levels are a little bit higher here. And then these cells are dividing, and they're leaving the terminally differentiated cells in their wake. And there's nothing to, there's nothing to make these guys grow, they only die. And so if these guys separate too far from the neck region, then the cells in the neck will die, and that's how you separate.
Question?
Yes.
So I don't quite understand the spatial distribution. So you have essentially no terminally differentiated at the front, where you have the.
At the front, you have these peaks where you have the stem cells, and then right next to them you have the, I'm showing you the dominant. There's always going to be some terminally differentiated cells in here, but the fraction is lower. The higher fraction is to the side. So as these guys are proliferating, they're leaving behind the differentiated cells in their wake.
There's a more or less monotone gradient, small at the front of the inhibitor, if I look end to end.
So the inhibitor is being produced by these cells, and so it's going to be higher here than here. Yeah, and so that's why these guys are growing out.
The white is both dead cells and the water.
So the white is, I'm not showing you the dead cells, and the white is the host. So outside here is, and the white is the host. The blue is the tumor. And so basically what you have is the spatial structure. These you can think of as niches for the stem cells, and that's emerging because of the self-organization. And they're at the tumor boundary, and and so you have this invasive growth that's driven by the stem cells. And one can ask, well, is that, how realistic is that? So here's a picture of, this is an intestinal crypt. So this is an example from the colon. And so this is work from the Cleavers group, where basically from a single stem cell, they can actually grow a crypt in vitro. And then the green is a marker for the stem cells, basically for the Wnt signaling. And then it's right at the fingertips. And so it's hard to see with this light, but the tips of these fingers are driven by stem cells, and these are the proliferation markers. So most of the proliferation is occurring at the tips of this guy. If we take a slice of our 3D simulation, then this is what it looks like. So again, everything is uniformly distributed at the beginning, but then because of this pattern-forming system, you get regions where the self-renewal becomes high, and so that's where this positive factor is being produced highest. And then you'll form these clusters right around where these guys, where this self-renewal factor starts to, the patterns start to emerge, so we get these patterns, and these guys are driving the invasive fingers. Okay, so
how do you select the
and quite a range of parameters. So this, in general, the system is, once you're into the Turing regime for that pattern-forming system, it's very robust. So we'll always produce evolution that looks like this. Quantitatively might be a bit different, but qualitatively this is the generic behavior. There's a question of whether you should be in the Turing regime at all, but we assume that you are. Yeah.
Why do they separate? Is the adhesion between cells very low, or?
So there is right. So there is the adhesion between cells is fairly low, and so what happens is that in these necks, this is mainly the differentiated cells, and so then these cells tend to die, and that's why, and they're not being replenished as quickly because they're really only being replenished by these stem cells, and then the committed progenitors, which I'm not showing, are behind them. And so then the cells in these necks, since there's not very many stem cells in the center, there's nothing to resupply these cells, so they'll just eventually die. Yep.
Maybe we're saying that even though you have a cluster of cells that basically is staying organized and is moving in a given direction, that's happening because there's a nutrient gradient and you have cell birth and death.
Correct.
You don't need any kind of chemotaxis
That's right.
to get this movement.
That's right. So this is not active movement.
Yes.
That would be on top of this. Yeah, there was a question?
Yes, because I mean the proliferation and the Wnt signaling are correlated.
Correct.
Only in stem cells, and basically any cell.
Mm-hmm. But the stem cells here are the ones that have the high Wnt signaling.
But there's also the ones with the proliferation.
The ones that so these proliferating cells tend to be the ones with the higher Wnt signaling, or at least the ones that are connected to the Wnt signaling. And so for our model, what we're using is Wnt signal as a marker for it. It affects both the proliferation of the stem and the committed progenitor. And so then our proliferation is localized around where the Wnt is high. Okay?
Okay, well, we can discuss this later.
Okay. Okay. So, for our system, Wnt will drive proliferation and will drive self-renewal for all the cell types, but we've assumed that the terminally differentiated cells don't proliferate. So now that was for just to get our feet wet for the avascular system. Now we want to talk how will this tumor actually interact with its environment. And so the interaction with the environment is very complex. And so the tumor cells will interact with the immune system, so macrophages. It'll interact with stromal cells such as fibroblasts, pericytes, blood vessels, and the endothelial cells. And so what we want to do is start to use this as a framework for incorporating these types of interactions. And so I'm going to here focus on crosstalk with the vascular network. We've also done work where we've interacted with fibroblasts and are starting to work on macrophages. But one can use the framework to try to do that. And so I'm going to focus on the interaction between this tumor and the vascular system. And so basically, there's crosstalk, at least in the glioma and a variety of other tumors, where it's been observed that the cancer stem cells for gliomas, which can be marked by a variety of different markers. So this is one of the markers, CD133 I mentioned earlier is another one. And so what has been observed is that these cancer stem cells tend to be located near the capillaries. So here's an example. This green is indicating this Nestin plus cell, so the putative cancer stem cell, and this is a reconstruction of a capillary. So this is from an experiment a few years ago now from Calabrese. And so what they did is they actually quantified for a variety of different tumors and for cell types the distance between from the cell to the capillary. So this is zero distance, and this is distance, I can't read it from here, 100 microns or so, 150 microns from the capillary. And so in particular for glioblastoma, so this is the nestin plus, this is the nestin minus, so the nestin plus are the putative stem cells. So there's a much higher fraction of the cells overall near the blood vessels, near the capillaries. And so they asked, well, what's going on? And so what seems to be going on is that the endothelial cells are providing self-renewing factors to the tumor cells, and in particular to the tumor stem cells. And so here, basically, they looked in conditioned media where if they don't condition the media, or if they do condition the media with these endothelial conditioned media from these endothelial cells, they will produce the plus will produce larger tumors. So it was believed that it will self-sustain the tumor. So there's this crosstalk. So now not only are the stem cells themselves providing their positive factors, and the committed progenitors providing these positive factors, but the stromal cells, so in other words, in this case, the blood vessel cells, the vascular endothelial cells, they're also providing factors that upregulate self-renewal. And so there's been a variety of mechanisms. When that work first came out, they didn't know why, but now there's been a variety of mechanisms proposed to explain this that involve both soluble factors, so diffusible factors, as well as contact factors such as Notch. And so, for example, nitric oxide, hedgehog, and so on, that are soluble, that are being produced by the endothelial cells that are signaling to and providing self-renewing signals to the cancer stem cells. And so what we can do is then incorporate that in the model by introducing another factor. Again, that satisfies a reaction diffusion equation, where now the production is the production by the vascular endothelial cells, so the blood vessels.
But this is posed outside of the cancer cell.
Yes, right. So the source is from the blood vessels, right? So this is a factor that now is diffusing in the environment. If you didn't have any blood vessels, then you wouldn't have any of this factor. Or if you didn't have any endothelial cells, you wouldn't have any of this factor. So it's outside the tumor system. But it's coupled to the tumor system because it's going to be coupled through the blood vessels.
So, but your velocity is the same. You are assuming that you have the same velocity inside the tumor as well as in the vascular.
I'm assuming that all the cells are tightly packed, and so any type of cell will move with this mass average velocity, and then the liquid is moving with this other velocity. So again, that's just for simplicity, because I can write down a system I can solve. If we assume that there are different velocities, we can make some constitutive laws for how those different velocities may behave, but we don't really know how how that system will behave yet. That's a much more complicated system to solve.
How will they interact with the boundary?
So essentially what we're assuming is that the host cells are moving with so basically you have proliferation events which are creating pressure that are moving cells. And so the host cells are subject, and the host stroma is subject to that same velocity, but there's no proliferation outside. And so then the velocities will be different, but they'll be continuous at the tumor boundary.
Just coming back to your earlier simulation. In your earlier simulation, you showed that a nutrient gradient could give rise to an asymmetry where you had selectively CSCs at the high nutrient end of the cluster.
Yeah.
When we do agent-based simulations, you don't need to have a support factor from the vasculature. The fact that the vasculature is producing nutrient of whatever kind is sufficient to give those kind of gradients where the stem cells stay preferentially at the surface.
Right.
Depends a little bit on how you do your advection and other things. But does your model, if you include vasculature, do you need to have this additional stem cell promotion factor, or does it work even without that just because of the nutrient gradient?
So what we assumed is that the self-renewal, well, basically the production of this factor depends on the nutrient levels. And so you would have to have some nutrient in the system. So in the avascular case, we just assumed that the nutrient outside the tumor is roughly uniform, so given by an underlying vasculature. And so you wouldn't need any.
So you have a factor that's specifically supporting CSC to CSC.
Yes.
But what I'm asking is, if you simply have the nutrient gradient because nutrient is coming from the vessels, in some models, versions of your model, you'll get the stem cells preferentially next to the vasculature simply because there's more nutrient there.
Yes.
So you don't necessarily need a second factor that's CSC specific.
I see what you mean. You mean, why do I have to have this factor at all?
Right. In other words, the nutrient gradient by itself will give you a preference of having stem cells
Yes.
next to vasculature.
Yes. That will, right, but this will be on top of that. Yeah. The reason for doing this is that we found that the increase is not that substantial just from the nutrient. Yeah. Okay. All right. So then what this additional factor does is just enhances the positive renewal, and this is one simple way of modeling it. And it also can affect the proliferation rate. And so next, what we have to do is I have to tell you, well, how do we get the vessels and how do we couple that with the tumor? And so for the so basically what is observed, of course, for vasculature is that in normal situations, the vasculature is nicely distributed, hierarchically distributed. But in tumors, it can be very irregular. And so what we do is we just use there's a variety of models in the literature. We just picked one that we had developed a few years ago, where we are tracking the vessels as discrete quantities, and we're going to keep the continuum representation for the tumor. So what we do in the angiogenesis model is we'll create sprouts from an underlying vascular system, and then we'll track the tip of these capillaries that form in response to various growth factors, pro-angiogenic factors, for which this vascular endothelial cell growth factor is a typical one. And then these the endothelial cells will chemotax up gradients of these pro-angiogenic factors, and then add probabilistic, so this is a probabilistic model, stochastic model, so with a probability, the tip can split and form new sprouts with probability, a certain probability that can also merge with an existing sprout. And so if we get sprouts that are connecting two, essentially two roots, so two parts of the vascular system, this will generally create pressure gradient, and then we'll get flow across this guy. So that would be pressure gradient in the vascular system. And so once you have flow, then you can have release of nutrients. And now the connection with the tumor is through that nutrient release and through the production of this factor, pro-angiogenic factor. So in a simple model, we can assume that cells that are feeling nutrient stress, so the nutrient is less than some threshold, start producing this factor, and then that distribution then triggers the angiogenesis, so the development of these new blood vessel systems. But then as this system develops, it can create production of nutrients by having flow that can then go back and affect how the which cells are producing the growth factor, and so the whole system is nonlinearly coupled. And so then to track these capillary cells, we just use we don't use a grid method, we just use a circular random walk model. And so now one can introduce this coupling to the tumor, and then in addition, these cells are also going to interact through the tumor through this extra growth factor we've introduced.
Is this in 3D?
Yeah. So again, we're going to take the same initial condition. So this perturbed circle, same parameters as with the avascular model, but now we're going to allow blood vessels to appear at a certain point. So basically when the concentration of the proangiogenic factors becomes high enough. So it'll be kind of hard to see because the image is going to get complicated, but the red dots will be the sprouts, the roots, where the vascular starts. I'm not showing you the underlying vasculature, just the new vessels that form. The gray will be the sprouts, so the parts that aren't producing nutrients, and then the blue will be the functional vessels. But as you'll see, it gets kind of complicated, so it's hard to see. So the evolution until the vasculature forms is the same as before, but now there's the vascular starts forming, there are the roots, the blood vessels, and now we only did it for a relatively short time, but this is now the tumor grown as vasculature. Now, what you can do is compare that to the case when it was avascular. And so it's quite a bit more fingered if we compare at the same time, and this is also quite a bit smaller. And so the vasculature is appearing near the tumor boundary, and it grows toward the tumor, and the tumor grows towards it. And you can kind of see here, there's a now stem cell cluster in the center because now we vascularized the tumor center with the angiogenesis. And as I mentioned, the fingers are less pronounced. And so we can analyze the differences. So for example, we can look at the measure a factor of the shape. So essentially measure the deviation of the shape from a sphere. So 1 would be a sphere, and increasing from 1 means that the surface area to volume ratio is bigger than what it would be for a sphere. It's appropriately scaled, so it's size independent. So this is that shape factor as a function of time, and so the avascular tumor has a much larger shape factor, so it's much more fingered. The vascularized tumor is much more compact. In fact, the shape is roughly stabilized here. You can look at the volume. So the volume, this is volume as a function of time. And so the vascular tumor, because now you're providing nutrient with the vasculature, grows much more rapidly than the avascular. And we can look at the fraction of stem cells, these cancer stem cells, and there, because of the positive feedback between the vessels and the tumor, the fraction increases by quite a bit. And mainly because now, in addition to having the stem cells at the boundary, there's this cluster of stem cells in the interior that are fed and supported by the blood vessels. And then this is just slices showing you what's going on. So this is the positive factor produced by the vessels. So the vessels are penetrating the center of the tumor, and so we have this factor that increases the so this is at an earlier time and the last time shown. And so you have the clusters at the boundary, but you also have this is the self-renewal. So the self-renewal is high at the boundary with the clusters, but also in the center where you get this positive factor that we didn't have before without the vasculature. And then the proliferation is increased in here. And then we have an increase in the fraction of stem cells in the center in addition to the ones at the fingers. Okay, so yes, question?
So this vascularization is here described as stochastic?
Yes, that's stochastic.
What is the actual distribution?
The actual distribution of what?
Distribution of this in this curve. I mean, this curve is for a single person.
Oh yes. So if we didn't calculate error bars for this, but our repeated simulations show the same trend. But I didn't calculate error bars. But yes, it's stochastic, and so in principle, I should run it again and again and again. But all our simulations behave in this qualitative way. Yes?
Are you assuming that the tips that are coming into the tumor eventually connect with each other to form a connected vasculature?
They may or may not. It's probabilistic. So if they have to get close enough, and then if they're close enough with a certain probability, they'll merge, or they may not.
They're the only ones that are used to provide nutrients.
They're the only ones that are used to provide nutrients. but we assume that any endothelial cell can provide this positive factor. And which brings me to my next point. So that's a simple model of the vascularization, and that's angiogenesis, the development of a new vessel system from the pre-existing. But what's also known is that the neovascularization, so the development of a tumor vascular system, is not just solely depending on the sprouting angiogenesis, the version I just told you, but actually can also be through vasculogenesis, where the most common form is that one can have vascular mimicry, where the tumor cells mimic the vascular endothelial cells and can line the blood vessels, and so that now the blood vessels can be mosaic. But there's also evidence that the cancer stem cells can actually differentiate into endothelial cells that have all the right markers and display all the right properties. And so those cancer stem cells, or those endothelial cells, cancer-derived endothelial cells, can also line the blood vessels. And so what's been observed is that the blood vessel system may be not just from the host, but also can be mosaic. And so the evidence for that different, they call trans-differentiation, going from the cancer stem cell to the endothelial cell, was observed in a set of experiments that now have been reproduced by a number of groups, where basically what they did is the following. They implanted a human glioblastoma into a mouse, and so they did it with cancer stem cells, and they did it with cell lines that don't have cancer stem cells. And then what they observed is that in the systems where you had cancer stem cells, by selectively staining for human endothelial cells, so human CD31, that's the endothelial cell marker, they could observe that there were blood vessels, or at least endothelial cells, and then they later showed they formed blood vessels, with the human CD31 that could only have arisen because they took the human into the mouse, could only have the human tumor origin. So here there's the staining for both human and the murine, the mouse CD31, so you see the blood vessels, but you don't see them in this non-cancer stem cells, but you see them when you do have the cancer stem cells, and this is staining for just the human. So this is human in mouse, and this is just human. So these guys have the blood vessels. And what they then, upon further analysis, they found that actually 70% or up to 70% of the functional vessels in the inner part of the tumor were human origin. And so that's in the core. But at the boundary of the tumor, most of the vessels were more mouse origin. And then that led to the investigation of, well, what's going on? And so here what they took is basically those glioma stem cells, cancer stem cells, and they put them in conditions that would promote endothelial cells. And so what they found is that if you take a patient-derived tumor and you put it under this guy, the cells start to form networks looking just like, well, qualitatively, like the human vascular cells. And then if you don't have the differentiated cells, they don't form these vascular networks. Then they did some further analysis and showed that you produce, for example, the endothelial nitric oxide synthase. So that's one of the factors that's upregulating self-renewal of the cancer stem cells. And they also showed that the vessels derived this way can be functional. So they can flow blood. So now, to incorporate that, we thought, well, let's take the model now and allow a branch to occur. So instead of having the cancer stem cells just go down what we would think of maybe as the glial type lineage in the cancer, they can branch and decide, well, maybe they want to become vascular cells. And so what we did is now said, okay, now to introduce a branch in the model. And so this is the model we had before, and now we can introduce a branching with probability R that the stem cells, instead of going down this way, can actually become endothelial cells. And so then a simple way of modeling that, again, being very simple, we can just now introduce a volume fraction for these tumor-derived endothelial cells. So here we're sticking with continuum. One could develop a hybrid method where we track these discretely also, but this just for simplicity. And so now we have a similar equation for these guys. They're, again, being moved by the same velocity. They're feeling the same type of flux. And now their proliferation depends on this branching probability. And then, of course, we have to adjust the the progression down this lineage according to the branching probability, and that's adjusting the source for the CPs, the committed progenitors.
Transient amplifying phase, or do you go directly from stem cells?
Here we just assume we go directly from stem cells, but it's not really known at all whether you could go from here or from here, so we just chose that. I think you'd get similar results if you went this way too. Okay, and then what we assume is that, well, these are bona fide endothelial cells, so they're probably producing this positive factor. And so we now introduce another source of this positive factor that's affecting the stem cell self-renewal from these glioma-derived endothelial cells. So now you put that in the model, and we're going to see that now I'm going to show you the vascular system, but now what's going to appear in yellow are these tumor-derived vascular endothelial cells. So now again, the simulation, the evolution is going to be the same as before until the blood vessels form, but now you see the yellow. So that's, the yellow is these, and then it kind of mixes with the green. So the yellow is the endothelial cells, and you can see that they naturally form a network inside the tumor. And if we take, this is just showing the endothelial cells. We take slices, you can see these clusters that are connected. The levels are fairly low. But you can do some statistics on them. And so if you look at in what we're calling the hypoxic core, so the region where the tumor cells are producing these angiogenic factors, most of these endothelial cells, glioma-derived endothelial cells, are in that hypoxic core, analogously to what's found in the experiment. And now we can look at some of the statistics. So the statistics look like this. So the simulation I just showed is this red curve. So this is the volume as a function of time. And so the green is the one, the green curve is the one that I showed you at the beginning with the angiogenic, just the coupling between the angiogenic system and the tumor. And this one is the one I showed you just now, where we have the yellow with the glioma-derived endothelial cells. And so these glioma-derived endothelial cells dramatically enhance the tumor growth. And that's because they're providing these positive factors to the stem cells. If we look at the volume fraction of the stem cell, you see that up here. So you can compare the red to the green. So a dramatic increase in the stem cell fraction. But if you look at the shape factor, so the deviation from the sphere, even though this tumor was growing the fastest, it's most sphere-like. And that's generically true. The less vascularized, the more invasive it's going to be, at least locally. Okay. So just to kind of put things together, this is the picture we've now modeled. So we have the vascular network. We have the that's coupled to the tumor by the tumor cells becoming hypoxic, producing these growth factors, telling the network to evolve. And then these network is affecting is upregulating the stem cells through this factor, this growth factor that we introduced. But at the same time, the stem cells can differentiate both into this direction and also this direction. And then these cells also provide this positive factor to the stem cells. So now that we've incorporated all of this, and what can you use it for? 5 minutes? Yeah. So in 5 minutes, I'll tell you what we can use it for. basically therapy. So we can now have this as a basic model to test the effect of therapies. And so since I have 5 minutes, I'm going to jump to the most interesting part. But you know, what can you do? You can do anti-angiogenic therapy. So what does that mean? It means blocking the blood vessels. It turns out these guys, so that typically the anti-angiogenic therapy is something targeting VEGF, the pro-angiogenic factor. Turns out these guys, because they have a different origin, are not dependent on VEGF. So traditional anti-angiogenic therapies will not hit those cells. You could do chemotherapy, or chemotherapy basically you're going to target the cells. You can do anti-stem cell therapy. So you can either kill the cells or you can try to reduce this positive factor. And you could do that by introducing its inhibitor. You could also affect the stem cells by trying to reduce its self-renewal by doing a differentiation type therapy. Oh, I think I just lost that. You can target the trans-differentiation, so you can try to kill those guys, and that would affect the influence of these guys on the stem cells. And you could do combinations where you try to target everything. So let me jump to the interesting case. Okay. So one thing you may observe, if you remember, is that when we got rid of the blood vessels, we had the tumor growing avascularly. The tumor became quite invasive. We had these fingers that developed, and eventually they might break off or not. And so here's an example of what happens. This is with chemotherapy, but if we combine it with anti-angiogenic therapy, what happens is that the tumors will grow the tumor will grow less. The control is this blue where you don't do anything. The tumors will grow less, but they become more invasive. If you look at the shape factors, the shape factors go up.
There's a stick just by the corner somewhere. Should be.
Oh yeah, thanks. Yeah, this is reliable. So so now you could ask, how can you target invasiveness? And so since the stem cells lead the finger development, by targeting the stem cells, you can also target invasiveness. And so one of the ways of doing that is enhancing the inhibitor. So we can imagine that we are going to provide the inhibitor from the blood vessel system. And so now that's going to be the result. So now we're providing the inhibitor, so we get rid of the stem cells at the tips. But now notice what happens. The whole thing is yellow. And if you see there's the red inside. We're not able to get at the stem cells in the center because this negative factor doesn't make it all the way in. And what's actually happening is that these endothelial cells, these tumor-derived endothelial cells, are protecting those stem cells. They're enhancing resistance. And so, so in particular, what you see is that even though you reduce the size, depending on the levels, this is different production. So this is the shape factor. Maybe I should show in terms of volume. This is the production level of that inhibitor, essentially the level in the bloodstream. And so this is volume as a function of time. So with the biggest value we used, we get something here. The tumor will eventually grow, but it's growing nice and compact. If you look in the literature, you can find examples of this. So here, these guys were inhibiting the Wnt pathway. And so the control is this black. And then here, they're inhibiting at different concentrations, basically different concentrations of drug. They can reduce the tumor, but it will still grow. It's quite analogous to what we're observing here. And then we have this additional factor of shape. And this is the, and so again, this is the images, the volume fraction of stem cells. So the highest concentration was this guy. So we just had, that's the simulation I showed, we just have this ball in the interior. And if you take slices, essentially what's happening is that this inhibitor is not penetrating the center. These are the stem cells, so you have this blob of stem cells, and the endothelial cells now end up surrounding the whole guy, and they're insulating it from the treatment, and they're providing the positive factor. Differentiation therapy does something similar. Let me skip on to the key. So now, if we focus on these cells are insulating the tumor, and they're providing this source of positive feedback that's maintaining the stem cell cluster in the center, and that's preventing the tumor from being eradicated. They're giving resistance. And so just the simulation I just showed, basically the tumor, this is so you see that tumor again with the stem cell cluster inside, and it's still continuing to grow. So now, suppose that we are somehow able to target these endothelial cells. There are, you can ask me at the end, there are some therapies that seem to be able to do this, at least from our looking in the literature. And so now, let's ask what happens. So what we're going to do, we've targeted the blood vessels, so we're getting rid of blood vessels. We are going to apply the Wnt inhibitor, so we're doing the anti-stem cell therapy by inhibiting the Wnt, so blocking the Wnt pathway. And now we're going to combine that with this, treating the endothelial cells. All the other parameters are the same. We now just added this additional treatment. So now notice we got rid of the yellow because we treated and got rid of the endothelial cells. And now look what happens. Then the anti-Wnt works. You get rid of this cluster, and then the tumor, at this point, there's not enough stem cells to sustain it, and the tumor will vanish. And so this is just showing what happens to those glioma endothelial cells. We're able to target them and get them down. And what we find, independent of what type of treatment we give, we find that these glioma-derived endothelial cells are enhancing the resistance against any type of treatment. And there's evidence in the literature that indeed that is happening, and that's the conventional belief, but then there's an experiment here that at least provides some confirmation of that conventional belief. So here, this is a measure of the apoptotic index, so essentially cell death. And so this is glioblastoma cells if they don't do anything, so there's some background level of apoptosis. When they treat them with, so this is radiation. I was talking about chemotherapy, these anti-therapies, but the results, I believe, should hold for that type. It'll be very similar. So if you apply 5 grays of radiation to the glioma cells, you dramatically increase death, so increase the apoptotic index. If you now mix endothelial cells with the glioblastoma cells, you don't do anything. You have this background level of apoptosis, and now you apply 5 grays. So that would ordinarily do this, except when you mix with endothelial cells, you don't get any change. So this is, we believe, evidence in the literature for this increased resistance. And so now the idea here is that one should combine, so to treat the tumor, you're looking for the Achilles heel. So here you do everything you can, you think, to affect the stem cells, but unless you can treat the Achilles heel, which in this case was these endothelial cells that were emerging from the tumor, stem cells, they provide the additional resistance. So to get rid of it, you have to target those as well. And so in the interest of time, let me stop and take any other questions.
Yeah, thank you very much, John. We have time for 1-2 questions. Come off over lunch, I guess.
So I'm reminiscing about the talk right before yours and wondering, so how sensitive are your results to parameters in your model?
So we didn't do a sensitivity analysis, which now is clear we should do. We are exactly in the case Alf is talking about. We have a super expensive model, so we want to do that in an optimized way. What I can say about that is our simple parameter variations find that that what I've shown you is qualitatively true as long as we're in the range of the Turing system, right? Underlying this, there was that Turing model for the Wnt and the inhibitor. So as long as we're staying in the pattern-forming part of the space for that, these results are very characteristic, and they're very characteristic of theory one can derive for these type of models. So for example, in earlier work, we showed that analytically that if you have avascular tumors, those will be locally much more highly invasive than if you have vascularized tumors. So you can show that analytically, and that's borne out here.
If I understood correctly, you said that the hypoxic effect on the tumor is that it encourages the network to proliferate. And according to my very limited knowledge, I believe the hypoxic effect also inhibits the total volume of the tumor as well, somehow from inside.
Yes, so
has that contribution been taken in?
Of course, but so we have proliferation as a function of nutrient level. So where there's low nutrient, there's going to be low proliferation. So while I said that there was the vasculogenesis part, the induced endothelial cells that tend to go into the hypoxic core, they get driven there by motion. They don't proliferate. The proliferation isn't taking place there. They're driven there by the fact that they're being produced on the boundary and driven inward. And then once they're there, they're not proliferating because the nutrient levels are too low.
Okay, I think everybody is hungry for lunch, and we can have more discussions there.
Yeah.
But let's thank John first again.